but by the time there was no national program, and I'm glad to see something going on. I would like, I'm sure Srivastava is much more in contact, and sometime during his lecture, he could tell us what is the status of those things in India and what we are about to do. Because the PO cities are becoming very popular, drug delivery systems. I am not sure how popular are drug delivery systems. One of my obsession was whether we can come up with a drug delivery system for diabetes. And a lot of people were attempting, and I would like Professor Srivastava to comment on some of these things. It will be more thing. And if he is doing such kind of work, I wish him all the luck, and it's time that we get some such things in India. All the best, Dr. Srivastava.
Thank you very much, sir.
Thank you, sir. Professor Mohan, would you like to say something?
Yeah, Dr. Ashok, I was very particular that I should hear this lecture because, as Dr. Atre mentioned, what are the latest developments in India as well as abroad. In fact, I also had this problem of diabetes. I could control it to a maximum extent. And I'm also aware that this is happening at IIT Madras also. I saw a lecture and I saw a startup company doing that. So how it is possible to control the diabetes? Is it the artificial intelligence apart from the MEMS devices, what we do? They are doing it with a sort of strap. Basically, they're trying to do those things. But I did not try that. I tried in my own way. Fortunately, I could control to a certain extent, reduce my tablets also. Very much interested. Apart from diabetes, what are the Dr. Rohit has already done a lot of things. I'm very much interested in hearing his lecture.
Thank you.
All the best, Dr. Rohit.
Professor Suresh, few words.
No, actually, I think just welcoming Prof Srivastava to ISSS, and we invite you to get involved in ISSS activities. Right? We look forward to your lecture today. Thank you very much.
Yeah.
Thank you.
Please, please, it's your time. Please feel free to present your lecture.
Sure.
Thank you.
Can I share my screen now?
Yes, please. Yes, please.
So, is this visible now?
Yeah.
Let me put it on full screen. Can you now see it on full screen?
Yes, Professor Rohit. Yeah.
Okay.
Thank
Is the
you.
slide changing? I'll just change one and show. Is that fine?
No, yeah, it is changing.
Okay.
Yes.
So let me start my presentation. Before I begin, let me thank all the stalwarts in the audience, and thank you very much for your kind words. I would like to begin this presentation by saying that we have done a lot of work in this area, but there is so much more to do, and the only reason I agree to give such talks at so many places is because I want to motivate everybody to keep doing what we have been able to do in a single lab at IIT Bombay, and I wish that every lab in every institute actually replicate what we have been able to do. So once again,
sorry to disturb you, Professor. Can you hide this front?
1 minute, yeah. Is it gone now? No.
Yeah. Oh, it's everything is gone.
Yeah. Now is it okay?
Yeah, now it is fine. Thank you.
So let me begin my talk by welcoming everybody to this talk. I come from IIT Bombay, and I started this lab at IIT Bombay about 17 years back now. 2005, I joined IIT Bombay. And since then, I've been working on many things. And I'm going to talk about my journey and where we are going from here, and where we see India going from where we have currently reached. So I start this lecture or any lecture by showing you this slide. And the reason I show you this slide is that I want you to be aware of the innovation ecosystem in your own institute. When I say IIT Bombay, since I come from IIT Bombay, I want to talk about IIT Bombay. But this is true for every engineering institute now in the country, which has connections to incubators, which has connections to medical colleges, which has connections to engineering colleges, which has connections to R&D laboratory, small and large companies in the city or around the city, and many departments within the institute. Now, let me give an example. If I were working in IIT Bombay, or I am working in IIT Bombay, in the Biosciences Bioengineering department, I have some science departments that help in many of my projects. But many of the things that I would need help with actually come from the engineering department. So there are many of these engineering departments like chemical engineering, electrical engineering, mechanical, industrial design, and so on and so forth, which are all working in this area. Now, let me now give you a broader example. Now, if I were to work on, say, a drug delivery project, and as Dr. Atre just mentioned, something for diabetes, I would need the know-how of drug delivery, which comes from biosciences. I would need the know-how of actually making a formulation, which would probably require my expertise as well as chemistry expertise. I would also need the know-how of somebody from mechanical engineering or electrical engineering to make a smart device to actually be able to deliver the device using a MEMS design. And then I would need somebody to program that so that it can become a smart device. So I would need somebody from computer science, or for that matter, even electrical engineering. There would also be the need of an industrial designer who would then design the entire thing so that we see what we would really like to see, and the users would be happy seeing that. Now, just this won't help us. I would be able to make a proof of concept if I was now in touch with some of these entrepreneurship channels inside the campus, something like an incubator on campus like Sign, or you have IIT Hyderabad has their own incubator, which is run very well, with many successful companies now. I would also require their support in putting me in touch with medical institutes, with engineering colleges, getting me private funding, because many of this, government funding may not be apt for it. So BIRAC funding is something that I would really encourage many of you to apply for. And then getting the different labs again now connected to each other so that we are able to take that proof of concept to a prototype stage. Once you get to a prototype stage, whether you can actually make an industry interested to take that idea from the prototype to, say, a manufacturable stage. At that point, you either tech transfer the entire thing to a medtech industry. I'll show you examples of that as well. Or you ask your students or encourage your students to set up a startup company, which we have been able to do very well. So, more than, 15 students in my lab have set up companies now. And we've got several tech transfers happen in the last 15 years or 10 years, I would say, not 15, 10 years. And that has now gotten, multiplied by many numbers, because, we are able to do it so well with many of these companies. And of course, in the process, now you're going to create these indigenous products, you're going to create affordable healthcare technologies, you're also going to create high value jobs as well as do a capacity building. Our department at IIT Bombay has several faculty, high impact faculty working on many things. But what I am interested in today is translational research. If each one of you or each one of us can work on at least one technology, imagine how many technologies we are contributing to India. And even today, we still are many technologies short, because the Honorable Prime Minister has given I think a goal of making 70% of indigenized technologies in India. So then I go to this slide, and this slide I don't spend much time. My only reason for showing you this slide is, if you look at this one slide on the top right hand corner, you see health expenditure per person. And you see where India is compared to Brazil, compared to Russia, compared to South Africa. Countries which really we feel, we are much better than them, but they spend much more per person in health expenditure. And for India, 70% of that money that you see there is private spending. So if I make a 100 rupee product, and imagine, I'm just talking of 100 rupee. If I make a 100 rupee product, it's not going to sell in India. Nobody is going to buy that in bulk in India, because people still are earning 500 rupees per day as laborers, as daily wage laborers, and maybe 800 per day as skilled laborers. If these are the people who are going to use many of the technologies or many of the products that me and you make, we have to make it cheaper for these people. So with that goal in mind, our whole journey started with affordability. And we said, anything that we do and any technology we make, it has to be cheap, and it has to be under under say $0.50 or 30 rupees or 35 rupees, anything less than that, then you start seeing an impact of that. I would say 10 rupees, but then there are some things that, we were not able to get under 10 rupees. So there are probable solutions. You see that on the left-hand corner. And these are, you make these affordable devices, you make early diagnostics using these technologies, you make them user-friendly so that, you have very little steps involved, and you make very simple reagents in storage conditions so that, you can earn money on the consumables rather than the device. And that's the goal for many of the technologies we made. Now, let me go to the next slide. So we started this journey in 2005, like I said, with many things that I learned in my PhD. And I came with a I came with an engineering background for my B.Tech. Then I went into biomedical engineering for a master's, which was completely MEMS. 2 years I worked in a class 100 clean room in the US. Then I shifted focus and worked for 3 more years in nanotechnology to earn my PhD degree. So I got both my master's and my PhD in 4.5 years in the US, working on 2 different areas, completely different areas, but which gave me a flavor of what biomedical engineering is. And the reason I show this slide to everybody is to tell you that biomedical engineering is not just one domain that you worked on in your master's or your PhD or your bachelor's. You have to think of the entire field. Are you able to contribute to the entire field? So you can work on drug delivery, you can work on tissue engineering, you can work on artificial organs, you can work on bioinformatics, you can work on artificial intelligence, you can work on point of care diagnostics or instrumentation, you can work on nanomedicine. All the areas are there. We've worked on all these areas. We started out doing very simple stuff in drug delivery, in hyperthermia, in cancer photothermal therapy. But then, within 5 years of my journey at IIT Bombay, I figured out that this is not the path I would like to go. If I go the same path as everybody else, I will end up at a certain point with so many papers, so many students graduated, but with no satisfaction of having delivered anything to this country. And I said, I want to do something different. 2010, I changed my outlook completely. I went into different areas. We started working on affordable paper-based diagnostics, on point-of-care diagnostic devices, on microfluidics, on biosensors, and many things that I had learned along with my students. And I said, I want to work on every new idea that our lab can work on and contribute in that area. With that, we expanded, kept on expanding for the next 7, 8, 10 years before COVID hit. And that also we considered a very big, thing for this country, because it's an opportunity. The country actually delivered. It actually delivered in PPE suits. It delivered in diagnostic assays. It delivered in reagent preparations. It delivered in masks. I don't know what else. So everywhere we thought we'll contribute. We started working in protective equipment. We've got 2 technology transfers in this area now from our lab. We started working in IoT-based health solutions. We've now got a large medical device company, Getting a Medical India Private Limited. It's a multinational that is working with us to create a smart ICU. We started working in disinfectants and sterilization products, in forensic diagnostics, in drug-eluting devices, in smart dressing materials. We just got a couple of big technology transfers in these areas and many, many other areas. So we've not left a single area behind, because we feel that if we can bring 1 or 2 technologies in each one of these areas, it will actually get excited for many, many people to start working in this area. Now let me start my journey. So the journey started many, many years ago, like I said, in 2010, when one of my students thought of making a product for the lab. And he said, sir, I don't want to work on a basic project. I want to work on a translational project. And I said, look, let's do that. Let's first figure out what we would like to deliver. First figure out the problem statement, the need, and then we start working on a solution. Now, the companies that you see in the bottom, on the top, on the right side, are the companies that started out from my lab. Some of them, and believe me, the Bioscience Technologies Private Limited started this whole journey, is now worth more than 1,000 crores. The Dynasense, the CareNX, the Neodox, the Norditech, have all reached more than 100 crore valuations. all of these companies are making big marks in this country just because they started out doing 1 or 2 products and then they expanded out into other products as well from the lab. So we've won many awards and we started doing many things with these companies. Now let me give you the journey. So the journey started with the one device in the center, which is called UCheck. And this UCheck is a mobile urine-based analyzer. This is the first instance of a mobile phone being used for diagnostics way back in 2010 in India. And this started the entire focus on mobile phones being used for diagnostics. And many such applications came into BIRAC. I was part of BIRAC panel. I looked at what the entire ecosystem was, and I have been part of BIRAC panel for now, I don't know, maybe 14, 13 years, since I started this journey, I've been part of BIRAC. And each year I see things growing. So, 1 year you will see many products come from AI and healthcare technologies. Then the other year you will see many technologies which are drone based. Then the other you see many technologies which are mobile based, and so on and so forth. So many things are happening. I'll give you some examples as we go along. The glucometer was the second from our lab because once we were able to commercialize one product, the second product was not very difficult, and we were able to do that as well. I'll show you the journey for that as well. But most importantly, our focus was affordability. So the UCheck in the center, we made sure that the strips were affordable. The device itself was just a mobile phone, and the entire thing that you see, it's a box. It's a simple box, a plastic box that doesn't cost much. All you have to do is mold it and make sure that you can give it for free with the mobile phone. The glucometer technology is very simple. The strip technology is a little complicated, but we made sure that we made it, we validated it, and then we started mass manufacturing using a contract manufacturer. And this was then available at a very low cost. I'll talk about cost. Somewhere around that time, Biosensors was also able to put their non-invasive hemoglobin monitoring system in the market, which we have used extensively. We worked on that, we used that extensively for many other things that I'll talk to you about. We also got several grants, and grants were one and plenty during that time. Today, all my grants have become industry focused. So I have maybe 1 or 2 grants from the government, but maybe 10, 12 grants from industry. And today our focus is industry deliverable. So the reason again I show you this one is the left-hand corner one, the Smart Sense, is an idea that we had pitched to Healthcare Innovation Technology World Cup. And this is a World Cup that is held every year by HIT Labs, New York. And this World Cup has participation of more than 170 countries. We participated, we put our idea, and this was just an idea. We went and presented it at New York and came third amongst the 170 countries. That brought a lot of respect for India. And most importantly, it got us into focus where we could now project this idea to Wellcome Trust. And Wellcome Trust was very happy looking at this idea. They put a team together and they actually put a mentor for us, Professor Nikhil Tandon from AIIMS New Delhi, who mentored us throughout the project, throughout the 3-year grant that this project was. And they gave us about £150,000 for that, but made sure that they gave us goals as to what they wanted the product to look like. I'll talk about that as well. And then we also did, projects for DST, for imprint, and many projects that we did over the years, all of them with a focus on commercialization. So our journey started with UCheck. And the reason I again tell you this journey is I'm going to motivate all the youngsters in this panel. Of course, take blessings from all the seniors. But my goal is to make sure that the youngsters understand that they have to first do a market survey. They cannot work on a product without doing a market survey. And many of you are already doing that. I know that because I know how IIT Hyderabad's incubator with Renu John is working. They already do a market survey. You do that, you find out what the need is, find out what the solution is in the market at that point. Then find out what you can offer, which is better than the solution existing. If there is no solution, then so be it. You have a solution which will give you a lot of return. But if there is a solution like this one, this was urine analysis. None of us really sit back and think about urine analysis. We figured the problem, went to path labs, talked to many of them in the small cities, also talked to many of them in and around Mumbai, found out the problem that they cannot even afford a simple urine analysis system. and Siemens systems were 75,000 to a lakh each. You cannot imagine a small path lab which has 10, 20 assays being done a day to be able to afford that. They were not. They could not even afford the strips. In fact, they got strips. They were cutting those strips perpendicularly and using them twice. Can you imagine that? 10 parameter urine dipsticks with pads in the center, 10 of those pads, and you're cutting them and then using them, for 2 samples. How do you know that your assay is equally divided in the pad, and so on and so forth? So many problems came. We found a solution. We actually proposed a solution to these people. What if we give you strips at this much cost and we don't bind you to a reader? You can actually use the color analysis, or you can use a mobile phone for diagnostics. And they were very happy. We got this funded as an idea from DBT in the IYBA system, and we actually delivered to DBT at the end of 4 years. And we said that, look, this is the product. The product is you will have a mobile phone with you. You can use that for your own calling or whatever you want to do. But whenever you are taking a reading, please use the mobile phone for taking a reading. There's a built-in algorithm. That algorithm is nothing but color analysis, and it's all made on. It uses image-based CCD array sensor, and this is simple to use. It does not take time. It is reflectance measurement, and you will be able to get absolute values once you can do a very good correlation with the inbuilt curve. Once you do that, all you need to do is make sure that these people use it. So we had to actually make sure that the mobile phone was connected to the internet. We had to make sure that we gave them strips which were cheaper than what was there in the market, and these were then priced at 1/3 what Siemens was selling it. So we picked the price carefully. We knew that we could not beat Siemens in the volumes, but we could beat them in price. And we priced them at 30 rupees for the 10 parameter. We priced it at 80 rupees for the 10+2, which is the albumin and creatinine strips, to give us the ACR ratio. So if you can do that, then you can make a market for it. And we really made a big market. Bioscience made a very big market out of this technology. They were able to sell these readers to mobile labs, to small path labs. Of course, we were not targeting large hospitals. But then you have to know what your market is. You cannot make one product which fits everybody. You have to make sure that your market is known to you. Go to tier 2, 3 cities, tier 3 cities, and you know that you have a $1 billion market waiting for you there. So with that, we got a big success story, and we were able to take it forward. Then we were contacted by ICMR to start working on a self-monitoring blood glucose system. And ICMR gave us a goal in mind. They said, can you make strips for us which are only 2 rupees and make a reader for those strips? We said, look, we don't know, because 18 to 30 rupees each strip costs in the market at that time. And we said, we don't know, we'll be able to deliver or not deliver. But ICMR put me in that panel. They said, bring in all the like-minded people from across the country and let's see whether we can do it. So we brought in people, we advertised this, we made sure that we got a glucometer scheme running, and the deputy DG and the DG office was very, very prompt about it. And we made these people come together, put their ideas, and looked at it. Once we knew what the target was, we started looking into the literature. And you have to do that. You have to look at what patents exist in the literature. You have to make sure that you don't infringe upon patents. You have to make sure that you do not work on technologies that are heavily protected. And these are specific examples are Israeli technologies. They are so heavily protected that you will never be able to use them. So make sure that you don't use such technology. So this is a simple one. It's all there in the textbooks. I'm not using anything different. It's a glucose oxidase peroxidase method. Finally, I'm using a dye which is changing color, and that dye you can use 2 dyes, just to make it more sensitive to the variations that happen in the instrument or in the atmosphere, and then you are done. Make a reader around it, make strips for the reader, and since we already had technology to make our 10 parameter strips, we were able to make the single parameter strips or two pad strips for that matter. These were now tested, and they were actually tested at hospitals. Whenever you are doing a glucometer, you have to also do a Clark's error grid testing. So if you can get more than 95% of your readings to lie in zone A of the Clark's error grid, you know that you are good to go. And ICMR was supporting this. We went to the National Institute of Design. We actually got this approved through the DCGI. Once we got
Professor Srivastava,
Yes.
is this only for random glucose or HbA1c also?
Sir, we are also doing HbA1c, so that I'll come to in a short while.
Okay.
Yes, so this is for spot glucose. We also have technologies now which we can do the HbA1c, which is 3-month marker, and we are also doing a 3-week marker, which is glycated albumin. And I'll talk about all of them. So this one got released in the market, created a big impact. The ministry was very happy. Everybody was happy, but we could not price it at 2 rupees. We priced it at 5 rupees in the Indian market. And with these 5 rupee strips, as you can see, those bottles, they were priced at so 50 strip bottles to 50 rupees or 20 strip bottles at 100 rupees. And we knew that we had a big market in front of us. Of course, many of these large players started bringing us down because they knew that if this technology reached the market, it would not be good for them. So we worked on a different version of it, and that is finally what got commercialized in a very big manner, which is the electrochemical strip base. Around that time, Wellcome Trust funded this project, and we said we wanted to do a point-of-care diagnostic assay for sodium, potassium, chloride, bicarbonate, pH, glucose, urea, creatinine at point of care. Now, this was a technology which exists in large readers, but you have to send the blood to the path lab or to the hospital pathology laboratory, where it will take some time and then come back to the ICU. Now, if a patient is admitted in the ICU, the doctor does not have time. The treating doctor needs the result ASAP. And this is a good technology to have on bedside. The only thing is that somebody will have to do a spin down of the blood, and blood is already being taken out. All they need to do is spin it down using the built-in centrifuge that our device had, put it in the built-in cuvettes that you now see on this slide here. And these built-in cuvettes or the cuvettes were sold as different assays. So we had sodium, chloride, bicarbonate, pH, and so on and so forth. So all these assays are separately given. All one has to do, and this has a certain amount of assay in it, all one has to do is add a little bit of serum or the plasma to it. In this case, if you spin it down at a very high speed, you get plasma. If you spin it at a low speed, you won't get the plasma. But anyway, the fact is that you were able to work with this and get data that was in accordance with what the laboratory analyzers were doing. And we did it at KEM Hospital. We actually did all our analysis and validation at KEM and found terrific correlation, as you can see on this data. But most importantly, we were able to show that the device and the assay could be commercialized, and we transferred the technology out to Bioscience. At that point, this is the sad part of the story, and I always say this, that one should not work on too many technologies. With one company, Bioscience was taken over by a company in Goa, and then finally by Perkinelli. So they were actually taken over, and then this technology actually just did not go forward. Now we are reviving this, and we are giving the technology transfer to another company which is visiting us and has expressed interest in this technology. So we hope that we will get a point of care device to do all these analysis for the critical care patients, and that's the goal for this product. We also did a project for DST, and I was part of their panel as well. And we also looked at many technologies. Of course, when we did albumin creatinine, we knew that albumin creatinine could also be done using a hardware reader. So instead of a mobile-based reader, could we do a hardware-based reader? And we did that. We actually went through the entire process of validating the assay, of making the device around it. So all the instrumentation happens in our lab. Of course, some of the PCBs, either we make it in IIT, in the PCB manufacturing, or we send it outside. If there are multiple layers to be done, then we send it outside. But whichever way, you can actually make the device, you can design it. And in this case, it was not a very good design of that sort, but still it did its trick. And this Monday, there is a company that is visiting us who will look at all this tech, all the data that we have, and we will do a tech transfer to the company to take it forward. I'm not showing too much of data. I'm showing you, highlights so that everybody is interested to see what we have done. Now, this is where I get very interested. We did a big project for NP mass, and Dr. Atre is aware of that. And he was the chairman. He was part of the committee. When we got this funding from NP mass, where we did a silicon-based device. So this was the one that we did. I'll come to that. But this excited us, and if we were able to make the silicon micro needles, can we actually make polymer microneedles with enough strength to push them under the skin, in the skin, with enough strength that they will remain in the skin, dissolve away in the skin, and release the drug in the skin. That is a huge technology, and that has interested many people. We now have data which shows that these microneedles can be made in different sizes. We've got molds for making these microneedles out of PVA. We've also got molds for making them out of PLA. But we are trying to avoid all the technologies that 3M has their patents on, and working on a different material which we know can be patented. Now, if we can make these microneedles, load them with drug, they can easily stick to the skin, and we've got enough data now that shows that they have enough strength that they can be pushed into the skin. Once they do that, you just leave the microneedles in place and they will deliver their payload. Now, these are not good for a large volume of drug. So if, as Dr. Atre was saying, if somebody wants to do it for insulin delivery, for diabetes, then one needs a reservoir in place. Now, there could be a way of adding a reservoir to this, because all you have to do is add a PDMS reservoir at the top and make sure that you press the PDMS reservoir so that the drug can go in through the needles. But then you need hollow needles to do that. And that's the goal for the next project. So the next project was, we actually were doing it, and we started doing it for NP Mass. We showed them how we could make microneedles, in collaboration with IIT Bangalore and IIT Bombay. Because in those times, we did not have a DRI at IIT Bombay. And the only DRI we could use was the IIT Bangalore one. But now subsequently, we got our own DRI. We can do it here. And we actually made sure that we made these microneedle patches. But not just the patch. Remember, if you just make microneedles, you're not going to get what you want to, because it has to be connected. And that's where we made our own smart device. So this is where I wanted to show to Dr. Atre and the rest of the committee, and all the people in the committee or in the audience, that if you can think of applications, if you can think of taking one small component and then combining it with a smart wearable device, then there are so many opportunities that open us. Now let me show you what we could do. We could make these microneedles. Of course, the funding was initially from NP Mass. Then we got a Gates Foundation funding. Then we got many other people interested in this technology. We could make these microneedles, and these microneedles initially we made it only using RIE. But finally, we could make them using deep reactive ionization process. They were as small as the hypodermic 34 gauge needles, as strong as them, and they could easily deliver the drug because they had a hollow bore. With this design, we said we wanted to make a smart device. The device was such that it could be worn on the skin, on the hand. It is a watch-like device, and the microneedles are at the base of the device. If we could do that and demonstrate that this could be done for one drug, imagine the possibilities it opens up for many other drugs. And that's what we could do. We actually went ahead and made a prototype for it. So initial prototype was really large, as you can see. But that's because we did not use a micro pump. We used a standard standard pump, as one can see in the picture. But then we also use tubings and pumps and the design, and then an entire electronic circuitry to make it smart. We programmed the drug so that the drug could come out in a program, as you can see on the right hand corner, and you could actually deliver the drug at the press of a button. So this was a technology that created a lot of interest from many people. There are people interested in it, but we are hoping that we'll be able to do a tech transfer to somebody who's really interested in taking this forward. As Dr. Atre said, there is a lot of interest in such technologies for diabetes patients who don't have to prick themselves daily, who don't have to think about refilling their reservoir, because all you have to do is add an insulin bottle to that, and that's it. It will actually do entire thing in automation. So if we can do that, that will be great. So there are many things. This is what the final version looks like, and the final version is much more sleek. It has simple design, and it can easily be integrated along with a drug reservoir. So that's where it stands today. We have not really done much for it after that point. But somewhere along that point, like I said, we've been working on diabetes, we've been working on kidney diseases. But Professor Padmanabhan in one of the NASI meetings mentioned that Rohit, everybody works on this. I don't see people working on lipids. I don't see people working on cardiac markers and so on and so forth. Of course, around that time, cardiac markers, IIT Bombay had also done some. Professor Ram Gopal Rao's group had done a lot of work in cardiac markers. Now, we picked up that challenge. We said, can we do a point-of-care device for total cholesterol, HDL, LDL, and triglycerides? And we submitted a project to BIRAC. There's a disturbance somewhere. So we picked up this project, went to BIRAC, got this funding. BIRAC has two schemes for it. One scheme is to go as an AIR, which is Academic Innovation Research. You get 50 lakhs over 18 months. In that 18 months, you demonstrate TRL1 to TRL4, which is just about a clinical validation, academic clinical validation.
Noise coming.
Yeah, there's some noise coming from somewhere. Maybe people can mute themselves.
Mr. Jain, can you shut off your... I think it's Vitraj Jain's.
Yeah. So what we did was, we said we wanted to. So I was talking about the schemes. So this is for everybody's benefit. BIRAC has two schemes: an academic innovation research, which funds academic researchers like us over an 18-month period to go from a proof of principle, not a concept, proof of principle to a proof of concept, which has gone through an academic clinical validation. At that point, you go back to BIRAC with an industry partner and get a CRS grant, which is a contract research scheme grant, which gives you whatever money you ask for and you can justify, all in grant, but then with the industry partner taking it forward. And the industry partner has to make sure that it gets commercialized at the end of whatever time frame you give BIRAC. So we went through both the schemes. I'll tell you where we are. So the first one was very complicated because we said we wanted to do a paper-based colorimetric assay for total cholesterol, HDL, LDL, and triglycerides. Complicated assays, as you can see here, 3 different enzymes putting on paper, but we made it work. Then the HDL cholesterol, the LDL is calculated, and the triglycerides. All of them we were able to do that on paper. We were able to design our system 2 different devices because we said we don't want to make it just paper-based. That will make it only home care. We wanted to make it, even for laboratory care, and that is where we wanted to work on a different version, which was a small cuvette-based version, and that's the design that you see here. Once we could do that, then we had to test the stability of the assay, test the device, test the assay through the device. We were able to do both. We then tested it in KEM hospital with their samples and their reader, and this is what values we got. We get terrific values which are in perfect correlation, less than 10% variation, as you see. In some cases, just about 10, but that's it. 10% variation. We have beautiful data with the analyzer that KEM has and our point of care assay. If we are able to do that, we went back to BIRAC and got a CRS grant. Today, we are 12 months into the CRS grant, maybe 15 months into the CRS grant. We've got it for 30 months. We now have to start the clinical validation at 4 different hospitals, around 250 samples each, and make sure that we file the test license and as well as the manufacturing license at the end of this project to be able to manufacture. And this is where we are today. So we've made a reader for it, a commercial reader. We've made the assay for it. We've made the strip which contains the assay, and we've got the entire design ready. And it uses very little blood, of course, a little more than what I would imagine. If had it been 15, 20 microliter of blood, that would be great. But at this point, it uses a little more blood, 40 microliters. But it can do all of this at point of care. And we are, just about finishing the design for the liquid assay, and that would also then be subsequently tested in the hospital. Subsequently, we got a grant from ICMR because we said, like I said, cardiac markers, troponins are a big marker, big marker because they are sensitive and they can actually tell you the incidence of heart attack. If you can do that, they are high sensitive markers. If you can actually measure it at their sensitivity, now this is where we are struggling, and I have no qualms in telling this because, it's finally research. In some cases we were able to do that. In some cases it is difficult. So we're not able to get down to 1 nanogram per ml. And that's the concentration that typically we are seeing in heart attack patients. So we are doing hospital trials at Nenepoya Hospital, and in most of the samples that we've tested, in about 25 that we have tested, maybe 5 samples are above 10 but all of them, 20 of them are above or less than 10, and most of them are between 1 and 5 nanogram per ml. That is a difficult thing to do, and we are still struggling with a lateral flow assay, because this, we are saying that this can be a marker that can sit inside the ambulance. As soon as somebody gets a heart attack and is going to the hospital, can we actually do a point of care assay and tell the hospital or the clinician, treating clinician, that look, we can see a marker troponin I or troponin T in the blood. Now can you start treatment, because this is a direct indication of heart attack. I wish I had good data at this point, but we're still struggling. We are still trying to do that, and hopefully, we'll be able to solve this, because we have tied up with a very big lateral flow assay manufacturer now, who has started working on some of this assay, and hopefully we'll be able to do this. And we've got the design and all that ready. We've got a mobile reader which can read, and we are doing a comparison with a commercial reader to do all that. We are also working on a urine analyzer now, which is more for the industry, because the industry has expressed an interest to work with us to deliver them a urine analyzer, just like the one I showed you in the beginning, the mobile based, but this is hardware based. And we now are testing that urine analyzer before we actually file for a test license. We are also working on a 3-week marker, which is glycated albumin in serum. And this is a big marker today, because if somebody has gestational diabetes, this will actually show up very nicely in pregnant women. And this is something that I think will become a big market in this country, because if you can do spot glucose, you can do 3-month marker, which is A1C. You can also do a 3-week marker, which can tell you a lot about the diabetes status of a person. And we are doing it again in a small assay format. Again, I don't have too many results to show you at this point. We've done the assay, we've made the design, and we've now started doing the hospital validation. Many technologies we are working on. And this is just a snapshot of all that. At this point, my lab is working on 40 different technologies. And I keep working. So Dr. Atre asked me about A1C. So you see that in the center, Dr. Atre, the one that shows a lateral flow assay using, for A1C. We've made that assay. We've tested it out with blood. We've actually got terrific correlation with our reader, which is a mobile-based reader. And now we are starting to do the clinical validation. If everything looks good, we'll actually have the most sensitive, because this doesn't require that sort of a sensitivity, but most useful assay out there at a very, very cheap cost. Maybe less than 50 rupees, I would say. I'm just giving a number here, but it could be much less than that, because we are using very little amount of antibodies, and we are able to get the A1C values in the ranges that we require. So many things happening in the lab. We are also working on many other technologies. I'll show you some of the other technologies as well. But just for everybody's interest, there are many things that can be done. You look at liver. There are all these liver markers out there. Can you make a point of care device for liver markers? Can you make a point of care device for all the kidney markers? And there are many of those. One of my students is working on NGAL, and we have plans to commercialize it this year. And there are many things that we are doing, which I think has a lot of interest for this country, because many of these markers have not been worked on in this country. And each one of us can contribute at least one device or one technology to this country, and we can reduce that many number of devices or assays that get imported into the country. We can save a lot of money for this country. We also work on nano-enabled technologies. Now, I don't know whether I should spend too much time on this, but I'll just give you a 5-minute brief on this. See, I work in many domains. Like I said, I work in drug delivery. So we've got these drug-loaded nano-engineered sponges to fight periprosthetic infections. We wanted to set up a company around it, and we were setting up a startup when a big company approached us and licensed the technology out. So both these sponges have now been licensed to the company, and they're going to make it more than, I think, 1,000 crore business, over the next few years. The sponges for on-demand delivery, the nano-engineered biodegradable bone and tissue fixation devices, we've got that made. We set up a company with Professor Belare and Kunal Khanna and one of my students, and we are now looking for funding for that. We've also got setting up a company in the photothermal therapy in cancer. That's going to be a big technology, because what we did, and Arvind is there at IIT Hyderabad, has been huge. And I also show IIT Hyderabad as our main collaborator here, because without Arvind's work, this would not have been possible. And we've done that. We've actually shown many things. We've got a GMP being set up in our own department. this will be able to scale up these nano formulations. Remember, when you do a nano formulation, you can do it in lab, but you do need to show an industry scale up. If you can do the scale up in a GMP facility and then do the clinical trials with that, the technology value goes up exponentially. And that's the goal for this technology. At some point, we believe it will become a million, $100 million company or so. But today, we are still struggling to get a few people on board for this. It is a good technology because it uses near infrared laser to hit the gold coated liposomes, and these then heat up and kill the cancer cells while degrading themselves. They don't leave anything behind, and the cancer cells are also dead. With this technology, you can imagine how much benefit a patient can get, even in an outpatient department. That's the goal that we started out doing, did a lot of studies with ACTREC, with Abhijit Dey's group at ACTREC. And we have oncologists who are very interested in this technology, people who are actually willing to try this technology out. We are only waiting for the GMP to get set up, and then I think we are good to go into clinical trials. We've got terrific data from toxicity, long-term, short-term toxicity, all done in GLP facilities, and we've got data that shows many things. So we've got many of these stimuli-responsive liposomal theranostic solutions, which many people are now very interested in taking forward. We've worked on bio-resorbable bone screw materials. We've got these, made compared to the bio-RCI HA screws that you see at the bottom there, at the bottom left, which is Smith & Nephew screws. Those screws cost 15,000 rupees. Our screws, this is the solution that we have on the top left-hand corner. And these have been made using a mold, and these have been made with a design that does not infringe the patent. So ICMR has filed an Indian, a US, a Japanese, and several other country patents. Set up a company, and we are now taking this technology forward. First in animals, because we know there is a big veterinary market for such screws, which are biodegradable, and then in humans with due clinical testing. There's a lot of novelty around it. As you can see, we've got a mold, we've got an injection molding system to actually make these screws, and they've got the right strength to actually counter whatever the foreign screws are out there in the market. Imagine the market. This is $1 billion market just in this country. Imagine the market that it can have in various other countries. Many USPs of this screw, not even going to go through all of that. Orthopedic surgical site implant infection. So imagine the technologies that one lab has worked on, and think of what you can do with just a tie-up with a hospital, with a clinician, with a surgeon. And this came from a surgeon who says that, look, this happens during surgery. Can you make a solution for that? We worked on this, made an antibiotic loaded sponges, both with multiple, one with single drug, multiple drug, and we actually showed release of the drug through these sponges over a period of time. He gave us time. He said, can you do it over 3 weeks? Can you do it over 6 weeks? Can you do it over 8 weeks, 12 weeks? And I can just pick that sponge 3, 6, 3W, 6W, 8W, 12W, and put it in patients after my surgery is done. He says it can become $1 billion market in this country. We are very happy that this technology is now taken over by a company, and they are taking it forward into production. So there are many things that our lab is doing. We are working on smart devices. So since I have an electronics background, I don't stop doing that. I am also working on interferotic drug delivery system, on sonophoretic drug delivery system, and we've got projects in both these areas. And we are hopeful that one of these technologies will actually reach, a really big market in this country. We are working on oral maxillofacial surgeries, and we are working on gel foam sort of material. And there's a surgeon who works with us on this, a hospital that is tied up with this. We've got comparative with many products. And finally, let me talk to you 2 minutes about the social impact that our lab does. So this is all commercial. We've done a lot of commercial work. We've earned a lot of money in the lab for IIT Bombay. But have we been able to be socially responsible for this country? And that comes out from this lab. We say, pick up one big problem for this country, TB, malnutrition, whatever, diabetes, pick up one big problem for this country, or, heart diseases, and see how you can make a contribution in that area. We picked up malnutrition and maternal health. And many of my students, 2 students, have set up companies in this area. And we've got malnutrition because we have worked on a smart scale system. We've worked on an anemia detection system. We worked on a smart height measurement system. And all of this is from our lab, which is out there. And we've got we've got a full maternal healthcare monitoring kit from CareNX, which is a student company who's doing a PhD in my lab. And everything is commercialized. We're not using any non-commercial devices. This is not for research, this is for CSR. Once you do that, you can actually take these devices in the field, actually be able to do that. So this device, an Anganwadi worker can carry around and do a lot of tests over a single day. Everything gets uploaded to a server. Anywhere she sees, or the device sees, or the cloud server sees a high-risk pregnancy, is referred to a tertiary care setup, or a secondary, or a primary care setup. In this case, it will be a primary. So the primary care setup immediately will have somebody bring that patient, and many lives can be saved. We've done that. We've done pilots in Maharashtra and Rajasthan through our student companies, in slums in and around Mumbai, in Palghar district, in northeast corners of this country, in Sikkim, in many northeast regions. And we've got grants now from DBT for a center of excellence to do malnutrition screening in 2 different states in this country, Maharashtra and 2 different states in northeast. We've got it for Ahmednagar district in Maharashtra. We've got data for Touch HB pilot in Churu district in Rajasthan. And for the care mother, we've got huge success story. We've saved so many lives. But most importantly, we have actually made sure that all the things that we do are making an impact somewhere. With that, let me now start acknowledging all the grants that we have received. We've got grants from BIRAC, we've got grants from Bill and Melinda Gates, from Grand Challenges Canada, from Wellcome Trust, from Gates Foundation, from Imprint, from GYTI. Several years my students have got grants from awards from GYTI, from Samsung Innovation, from Stanford MedTech. So you see all the big names here, because this is the pride for India. If we have been able to be featured in some of these foreign grants, I think that's a big thing for this country. And I hope many of us are able to do that. Overall impact created by my lab, we have trained so far in 17 years, I have trained 35 PhD students. They have all graduated. 22 are doing their PhD with me, even today. I have 8 M.Techs doing their M.Techs master's projects with me, and we have graduated more than 90 M.Techs. We have got more than 200 publications. Most importantly, we train a lot of interns and project staffs, because these interns don't get the opportunity that IIT Bombay can give them. I give each one of them a recommendation letter to multiple colleges and make sure that they get admits from foreign colleges. They learn, they are part of the lab, they are part of books, they are part of patents, they are part of publications, and they are actually making it very big for this country. We train many innovators, we help many startup companies, we file many patents. So till date, my lab has filed 150 patents. One lab, 150 patents. And we keep filing every day. Even today, I have approved 2 on the online system. So by the end of this year, maybe 170. By next year, maybe 200. I don't know how many. The fact is, if each one of these patents were to be commercialized, then imagine how much money it brings back to an institute. And that's the goal for the lab. We are doing many commercializations now. We've done many, we are doing many. We've got many startups. We've created many high-value jobs. We've made sure that we have showcased all this to several people who matter: the Honorable Prime Minister, the Honorable Science and Technology Minister, the Honorable HRD Minister. Everybody has been impressed by what IIT Bombay has been able to do for this country. And I'm sure all the IITs are able to do the same. All the NITs are able to do the same. So this is where we stand today. We do a lot of outreach. We make sure that we train young students, 11 and 12 standard students. We make sure that they come to our booth that you see every year in TechFest, and we make sure that we showcase our technologies at many places. We have a huge partner network, because we don't believe in working all by ourselves. I have put some of the IITs there, but there are many others. Many places where my students have gone as faculty, where they are creating big impacts with many things that they are doing. NIPERs, IIT Indore, NIPER Hyderabad, NIPER Guwahati, NIPER Ahmedabad. I don't know how many colleges now. INST, and so many of these colleges which are not even there. I need to update this slide. There are many hospitals that we work with, many small, large companies that we work with, many startups that came out of my lab. That's what my lab looked like at certain point. We had more than 50 people. Even today, I have 45 people working in my lab. We have a big tie-up with Indian Navy, with DRDO, with several large organizations, and we believe in creating impact because we don't believe in taking projects which we cannot deliver. We will always deliver at whatever cost it may be. If it means that we spend a lot of effort with very little cost, we'll do that. If the partnership matters to us. But last of all, let me give my heartfelt gratitude to my mentors, and I'm sure many of you have mentors. Make sure you always tell them how much you owe your career to them or your path to them. And this is how I do that. I always make sure to do that. Professor Padmanabhan, who's my biggest mentor, for many people in this country, he's helped many of us youngsters in this country. Dr. Manju Sharma, who has been a terrific mentor to all the youngsters. Sitting in NASI, she has seen the entire landscape in this country, and she has been able to pick people from this country who have actually delivered. And I owe our success to Dr. Manju Sharma and Ms. Dr. Renu Swarup, who has been again a huge mentor to all of us, and she has supported me in every venture. Finally, thanks to all the faculty members, collaborators, my students, and all the startups that I have worked with, and I'm going to work with in the future, and all the companies that I'm working with. Thank you, IIT Bombay. Thank you to the organizers, and thank you to everyone.
Thank you, Professor Rohit Srivastava, for outstanding talk and sharing your very interesting results. Now, Ashok has some issue with the power, so that he asked me to take over.
Sure.
So I request the audience to ask the question. Just unmute your microphone or you type your question in chat box. I will read it.
Hi, sir. I will start. I don't have any questions as usual. Great talk, sir. I enjoyed. And I must say the audience that I have witnessed at least 20 of those technologies which sir showed today when I was working in his lab at IIT Bombay. One thing is, despite doing all his work, this is one of the coolest labs that one can get. So we never felt the pressure of working. That is another thing that I want to point out. Thank you, sir.
Thank you, Arvind. Thank you so much.
Just a few comments. Prof Srivastava, I'm glad to see. In fact, it made me nostalgic because about 25 to 30 years back, I talked about this. I'm glad to see something like the nanotoporosis and troponin monitoring and others are doing. As you may recall, I tried to get this chip done through Ram Mohan Rao at one stage, the troponin and myoglobulin and so on. So this wearable device, is it marketed now?
No, sir. Actually, we were not able to transfer the technology out.
What is the reason?
Sir, I think many people expressed concern that it would not be cost effective. I don't know why, but we were not able to show, because sir, the DRI process that we used at that point, it was expensive to use. IIT Bombay did not have it, and we went and used at IISc Bangalore, where we were charged very heavily for it. We were never able to justify the cost for that. And wherever we were asked, we were always asked, but how do we scale this up? How do we make a million pieces of this? And we had no answer to that.
Are POCTs being marketed in India?
For what, sir?
POCTs are being marketed in India?
Yes, sir. Yes, sir.
On average, for what kind of selection? For example, the assay of the cholesterols or...
Sir, we were able to market the urine strip. We were able to market the glucose strip. And the cholesterol one is just about getting technology transferred now. So all this, sir, there are a lot of companies which are interested in taking the POCTs. But wherever the MEMS came into play, I don't know why the companies stopped discussing. Which is why we started working on the polymer based, which does not use a MEMS process, but it can actually use a dye or a mold to actually create these microneedles.
When will your drug delivery system undergo clinical trials?
Sir, so there are three different drug delivery technologies we are working on. One is the sponge based, which is the one which just got taken over by two companies, and both the companies are very interested in. So maybe by I'll say 1.5 years they have given us to complete all the preclinical, and they will put it into clinical trials at the end of 1.5 years. And the microneedle one, sir, we are still struggling to get a person to fund that. So we are not sure where that is going.
Just curiosity, one last question. Sure. You have looked at wearable systems and point of care. Anybody working with swallowable systems, injectable systems, and other things in India?
In what, sir? I missed the point.
Swallowable and injectable systems.
So injectable systems, there are many, sir, but swallowable, I have not seen anybody. Not anybody. So injectable, yes, there are many.
I think we should get our own endoscopic pill, you know.
Yes,
It's
sir.
very expensive.
I believe that, sir. Definitely.
5 to 7 lakhs, and I don't think anybody can afford it. I think we should look at how some of them. And one of the things probably we should do is have a consortium of all these biomedical POCTs and in India and exchange information. Is there a group like that in India? Is there do you biomedical people? Why don't you start it?
We can
the
start
interest group.
Definitely.
Because at least now, as you said, in many IITs and NITs, there is a biomedical group working. I think you should form an interest group and look at some of the problems so that you can share rather than working on the same system.
Yes, sir.
It's something which I would request you to lead and do that.
Definitely, sir.
Thank you.
Thank you, sir. Thank you very much.
Okay, any other question? Yes, yes, sir.
Rohit,
yes.
I appreciate your interest in translational research and especially the skill development programs for students. Being an academic, being a professor, training so many interns, so many students, and then motivating them is the best thing that you have done. The other part is basically commercialization, as Dr. Atra also was pointing out. What are the problems that you are facing and what are the solutions that you give? Because commercialization is most important. Doing so many devices, still not in the business in a big way. How do you solve the problem? Is there any advice from you to take it to these things to higher TRL levels, commercialization, and showing the market from India? What are your suggestions?
Definitely. I think that's a good question, Mohan. So my only advice would be to involve a consultant right in the beginning. So, whenever one does a project, there are two things. One is crucial, in the project. One is the clinician, which many people do. And now everybody is doing that. But people forget to involve a consultant who knows the regulatory pathway. And that regulatory pathway is the most crucial. If I can understand when to file the test license at CDSCO and what to do for that test license in order to get the clinical validation done at 3 different hospitals, and this consultant does the entire thing for a fee, and he will also make sure that he files the manufacturing license. At that point, there are at least 3-4 companies which will be willing to come forward and take the technology. What happens is that we always stop at academic clinical validation. We never file the test license. We never even file the manufacturing license. If we do not do that without the ISO facility, then we are never able to commercialize. So that's something that I'll tell everybody, if one can find a CDSCO consultant, I have known people, who work with me, but there are many people who struggle to find one. And that's where I think the biggest bottleneck is.
One indirect from what Mohan asked, you must have interacted with medical doctors. What is their attitude towards these POCTs and others rather than going to the standard laboratories and pharmaceutical things and so on? Are they open to these kind of things?
Yes, sir. Today, I think many doctors are very open. So we are working with Dr. Anurag Leela at KEM Hospital, and we proposed a point of care device for T3, T4, TSH, thyroid markers. And he is a very big endocrinologist at KEM, sees maybe 200 patients a day at KEM. He was so enthusiastic with the idea, he immediately put himself on as a co-investigator on the project and said he will do the clinical validation of all these that we do at KEM Hospital. So imagine, hospitals, which are government hospitals where doctors are actually interested in working on technologies with us and making sure that it reaches validation.
One of the things which should happen in direct answer to Professor Mohan's question is, if the engineers and scientists try to market biomedical project in India, it will be better if the clinicians try to market the professional project in India. There will be greater acceptance and immediate acceptance.
Yes, sir.
Because when a doctor says something, it comes from God's word.
You
and I say something, it's technological mesmerism or
right,
mythology and so on.
So in fact, BIRAC, you are very right, sir. BIRAC funds those projects which are led by doctors immediately. Rather, they ask a lot of questions which are led by engineers.
Okay, any other question? Okay, Professor Rohit, I
think
have
my
one question. Oh, sorry.
Yeah. Basically, I was asking with all the incubation centers at IISc, IIT Bombay, IIT Hyderabad, and many places, what is your suggestion to these incubation centers to solve this problem? What is that they have to do?
So they're already doing a lot of things now. Earlier these incubators were doing very little, but now these incubators have many programs. Where they are encouraging the young faculty, ideas, not even faculty, students to come to the incubators, pitch for 10 lakh rupees funding so that they can do a proof of principle. I don't think there is much
No,
we
Dr.
can do.
Rohit, my
Yes,
question is not encouraging incubating. It's solving the problem with industries. They have the money. Do you
yeah,
think that they have to have more money for the consultants?
sir.
Identification of consultants, paying for the
I,
consultants. What are your
Yes,
suggestions?
yes, sir. Most of them don't have empaneled consultants. So I think you are right, sir. If many of these incubators actually have empaneled consultants, then they will be able to help these incubatees much better, sir. Yes.
Good. Thank you.
Anyway, an excellent lecture, Rohit.
Thank you,
All
sir.
the best
Thank you very much.
down the road.
So, Professor Rohit, I have one question regarding this microneedles. So, the microneedles which you have fabricated looks like a cylindrical shape,
Right.
means a uniform dia.
Ah.
And generally, if you have the stable microneedle, it should be pointed, base should be wider than the top.
Yeah.
We struggled, sir.
Long back.
We struggled. But in fact, even with these, if one could press it into the skin, they actually go in very easily into the skin. They don't break. We actually did all those studies. So we struggled. We could not get the pointed needles, so we had to work with these, and they worked very nicely. So I would say that if one can make pointed, it's good. If not, we have to compromise somewhere.
But in case of the silicon, because silicon is not biodegradable,
Yes.
then can we use it?
Yeah. So silicon is biocompatible. So as long as you take it out, you don't leave traces of the needle inside the skin, it's fine. So
No, but it will some needle will break inside the skin.
so
It's
that's where the long-term effects.
We don't know. I think it's reasonably safe. Everywhere silicon is the one which is being tried.
Yes. I think Dr. Atre is much more versed in that, rather than me, with all this.
Okay. So any other question, please type in the chat box, or you can directly ask, just unmute your microphone.
Okay, I think looks like
If there is no question, then on the behalf of the audience, I would like to thank you. Thank you so much, Professor Rohit, for outstanding talk and sharing your all very interesting result. I think which, inspired us to work in this area, because I'm working in the area of men, so I know that some of the means devices are very interesting. So microneedle is just like that, much easier for me to make it pointed. So that's why I was asking the question, because I'm just working the wet bulk micro machining area. So making them sharp, just we have to give the wet etching treatment.
Very nice.
Yeah. Thank you so much.
Thank you
Professor.
so much. Thank you very much, sir. Thank you.
good night.
Right.
Okay, see you sometime, Mohan.
Good, sir. Thank you very much, sir. Nice to see you
Right.
here on this, at least.
Yeah. Okay.
We should attend more of these workshops, at least to say hello.
Yeah.
Thank you very much,
Right.
sir, for your all supports and cooperation. Thank you,
Good.
Mohan sir.
Good. Bye-bye, Dr. Paul.
Yeah, thank you. Thank you all.